Okay, so here we are at the end of this chapter. We talked about our porifera. We talked about our cnidarians. We talked about our lophotrochozoans, our platyhelminths, our rotifera, our mollusks, and our annelids. We talked about ecdysozoans. We talked about our nematodes and our arthropods. All of those beautiful arthropods we have. And now we are to our Deuterostomia. Our deuterostomes that we're going to study are the echinoderms, which are the invertebrates we're studying in this chapter, and then the chordates, which we have our entire chapter. Chapter 34 is all about our chordates. Our deuterostomes are animals that have radial cleavage. This is something I hope you remember from chapter 32, but if you don't, go back and look up what radial cleavage is. From the blastopore, that blastopore turns into the anus, and then the second opening turns into the mouth. That's what deuterostome means, a second mouth. There is an interocellous coelom formation. And mainly our deuterostomes are defined by DNA similarities, since we have a lot of other animals that share similar developmental characteristics. We have our echinoderms, which are the sea stars and the sea urchins that we're going to look at, and then the chordates, which are the vertebrates. Echinoderms tend to be slow-moving or sessile, so a lot of these maybe stay in place, but if they are able to move, they move rather slowly. They have a thin epidermis, and then the epidermis is covering an endoskeleton. So it's still not a regular bony skeleton like we have, but it is underneath the skin, and it is hard. It's calcified. There's calcium in it. One of the unique features of echinoderms is unique water vascular system. So if you think of echinoderms, I want you to think of a water vascular system. The water vascular system is a network of hydraulic canals that has a ring canal and radial canals. And the tube feet will function for both locomotion to help it move and for feeding. We usually have separate males and females and an external sexual reproduction. That means that the eggs are released, the sperm are released. There's no sexual coitus going on. It all occurs externally. Here's the tube feet of an echinoderm. Let's watch how it moves.
The sea star is a member of Phylum Echinodermata. Animals in this phylum have a distinctive mechanism of locomotion. Within the sea star is an internal water vascular system that terminates in branches called tube feet. As the sea star moves, it uses chemicals first to attach each tube foot to the substrate and then to release each tube foot from the substrate. Water enters the water vascular system via the madreporite, which is a strainer-like structure on the dorsal side of the animal. This video shows what happens when a sea star is placed on its back in an aquarium. The tip of the sea star's arm can bend back on itself, and once the tube feet contact the substrate, the animal slowly pulls itself onto its correct side. Credit courtesy of Graham R. Kent and Rebecca L. Turner, Smith College.
Okay, so let's review these animal groups we looked at in this chapter. Porifera, or sponges, they do not have tissues. They have choanocytes, which are those collar cells, those flagellated cells that ingest food particles. We looked at cnidarians, our hydas, jellies, sea anemones, and corals. These have nematocysts, those unique stinging structures, within their cnidocytes. They are diploblastic, they have radial symmetry, and they have a gastrovascular cavity. That is that little pouch, that compartment that has one opening that serves as both mouth and anus. Our lophotrochozoans, we have platyhelminths, which are our flatworms. They do not have a body cavity. They are acoelomates. They are dorsoventrally flattened. That means belly and back are flattened against each other. They have a gastrovascular cavity or no digestive tract. Like our tapeworms had no digestive tract. Our rotifera, we didn't look at these other ones, but our rotifera have a hemocoel. Rotifers have an alimentary canal. That's a digestive tube with a mouth and an anus. We looked at mollusks, which include clams, snails, and squids. They have a hemocoel, a reduced coelom. They have those three main body parts, a muscular foot, visceral mass, and mantle. Most of them have a hard shell made of calcium carbonate. Then our annelids are our segmented worms that have a true coelom. They have a body wall and internal organs that are segmented, except for their digestive tract, which is unsegmented. Our ecdysozoans, we looked at our nematodes, which would be roundworms, which have a hemocoel. They have a cylindrical body with tapered ends, no circulatory system, and they undergo ecdysis, or the shedding of their cuticle. We looked at arthropods, which included the spiders, the centipedes, the crustaceans, and the insects. They have a hemocoel. They have a reduced coelom. They have a segmented body, jointed appendages, that is a key feature of their clade, and an exoskeleton made of protein and chitin. Then our echinoderms, which include sea stars and sea urchins, have a coelom. They are bilaterally symmetrical as larvae and radially symmetrical as adults. They have a 5-part body organization as adults. They have a unique water vascular system and an endoskeleton underneath their skin. Then in chapter 34, our next chapter, we're going to look at our chordates, which will include lancelets, tunicates, and vertebrates. We have a coelom. Then the features of a chordates are having a notochord, a dorsal hollow nerve cord, pharyngeal slits, and a post-anal tail. All chordates have all 4 of these features at some point in development. Although you may not have them as an adult, you do have them at some point during development. Okay, that finishes up this chapter. Can you believe you made it all the way? This was a big chapter. And 34 is going to be another big chapter. Then we'll get to start studying our different animal body systems. And so we have some fun coming up this semester. All right, let me know if you have questions. I'm here for you when you need me. Bye.